Hi everybody, I hope you are all well and welcome to our lesson on genetics. This is Unit 4, it's our fourth lesson, and what I love about the topic of genetics is that it incorporates all the previous work that we've done. It links up DNA and RNA, and even the concept of meiosis plays a role in genetics. So when we think about the word genetics, we think about the word gene, and we first saw the word gene in our work under DNA. And we said that a gene is simply a segment of a chromosome which helps to code for a certain characteristic. When we moved on to the concept of RNA, we looked at how that protein is made. And when we looked at the concept of meiosis, we saw the process of gamete formation. And genetics links all of this into one topic. So let us now jump into this topic and see what it is all about. The first place where we will start off in genetics is looking at genes on a homologous chromosome pair. From our work on meiosis, we know that a chromosome pair is homologous if the chromosomes in this pair are the same size and if they contain the same location for their genes. The location of a gene on a chromosome is called a locus. And we can see that the gene for flower color is found on the same position in both of these chromosomes. So in chromosome number 1, we have the gene for flower color, and in chromosome number 2, we also have a gene for flower color. But most importantly, since they are homologous chromosome pairs, the locus is the same. In other words, the position of the gene on each chromosome is exactly the same. The gene is flower color. However, the expressive form of that gene may differ. Chromosome number 1 codes for a purple flower, whereas chromosome number 2 codes for a white flower. So although both of these genes code for a flower color, the expressiveness or the physical look of this gene may differ. It may be purple, or it may be white, or even a combination of both purple and white. And the different versions of these genes are called alleles. So, to summarize the language that I used, firstly, we are talking about a homologous pair of chromosomes. Secondly, a homologous chromosome pair is homologous if they have genes on the same positions. The position of a gene is called a locus. A gene is the recipe for a trait. However, there are different versions of the same recipe, and these are called alleles. So for example, if you are baking a cake, you may bake a chocolate cake or you may bake a vanilla cake. However, you are still following a recipe of baking. So in the same way, your recipe of baking is your gene. But if you are baking a brown cake, or if you are a brown or chocolate cake rather, or if you are baking a vanilla cake, that will refer to your alleles. And generally what we find is that one allele is dominant over the other allele. So if we make the assumption that purple color is dominant over white color, it means that when we have these two alleles in combination, purple color will be expressed in your flowers. Let us now introduce more terminology important to this topic. This includes the word heterozygous and homozygous. And these two words refer to the relationship that is found between your two alleles. In order to explain this, I've chosen to look at an example of fur color in dogs. So let's assume that a dog can have either brown fur or white fur. And furthermore, that fur color is controlled by a single gene. I've noted on the top that brown fur is dominant. When we want to represent the genetics behind fur color, we use letters of the alphabet. When we are talking about the alleles of a gene, we have to use the same letter. So fur color, whether it is white or whether it is brown, must be represented by the same letter. But the dominant allele is represented by the capital letter of that letter, and the recessive allele is represented as the lowercase letter of that letter. So let's assume that brown fur color, because it is dominant, is represented by the letter capital B. That then makes white fur color a small b. So in other words, capital B is dominant, it is brown fur color, and small b is recessive, it is white fur color. So let's come to the first concept, heterozygous. If a dog is heterozygous for fur color, it means that at a chromosomal level in a certain homologous chromosome pair, it contains an allele for white fur color and an allele for brown fur color. And so we remember that white is a small b and brown is a capital B. So when we represent the genotype for fur color in a heterozygous dog, it is capital B and lowercase b. We tend to write the capital letter first. In this combination, because B is dominant, B will be expressed, and that capital B is brown fur, so rather I should say the capital B will be expressed and brown fur will be expressed because it is dominant, whereas small b or white fur will be masked and it won't be expressed. Next, let us come to the concept of homozygous. And when we look at this homologous chromosome pair, we can see that we contain the allele for the dominant fur color on both chromosomes of your homologous chromosome pair. So we've got a capital B and a capital B. And the genotype in this instance will be capital B, capital B. We have two dominant alleles, and so our fur color will definitely be brown. In this instance, we can say that it is homozygous dominant. However, there is also a possibility that instead of having brown allele and brown allele, we can have white allele and white allele. And in that instance, we will have a small letter b and a small letter b. Remember, we have to keep the letters the same because we are talking about the same gene. So we can't use the letter W, for example, to represent white fur color and the letter B to represent brown fur color. Just keep that in mind. And in this instance, our genotype will be small letter b, small letter b. And so we will have a dog with white fur color. So some important concepts to remember. Heterozygous means that the two alleles are different. Homozygous means that the two alleles are the same. We can be homozygous dominant, as in this example, or we can be homozygous recessive, as in this example. We also make the assumption in genetics generally that a trait is controlled by a single homologous chromosome pair. Now let's see how the concept of meiosis links into genetics. So on this image, if we have a look at the top, we contain a heterozygous diploid cell from a plant with yellow seeds. And we can see that they represent that with the letter Y. Capital Y is dominant and small letter y is recessive. Obviously, since the seeds are yellow, we know that yellow has to be expressed, and thus in a heterozygous combination, yellow must be the dominant color. However, let us talk about Mendels' law of segregation. Mendels' law of segregation simply states that during the process of meiosis, your two alleles of a homologous chromosome pair will split up into individual alleles in a gamete. So if we have a look at this image here, we have two alleles, small y and large y, or lowercase y and uppercase y. Obviously, it is duplicated, so that is where the other uppercase y and small letter y comes from. But as the process of meiosis 1 and meiosis 2 takes place, your gametes will then be formed, and we can see that one gamete will have an uppercase y, another gamete will have an uppercase y, and another gamete will have a lowercase y, and your last gamete will have a lowercase y. So your two purple gametes have the allele for recessiveness, and your two yellow gametes have the allele for dominance. And in this instance, the trait we're talking about is seed color. So your purple gametes will have the recessive allele for seed color, and your yellow gametes will have the dominance allele for seed color. And through the process of fertilization, a dominant allele might meet up with another dominant allele, a recessive allele might meet up with another recessive allele, or a dominant allele might meet up with a recessive allele. And in that instance, we will then have a new seed color. But the point that I wish to emphasize is that through Mendels' law of segregation, we find that the two traits of a homologous chromosome pair split up during the process of meiosis, as so that each gamete only receives one of these alleles. Next, we can apply all of this knowledge into understanding genetic crosses. There are 4 examples of crosses we are going to look at, starting off with monohybrid crosses. Monohybrid crosses involves a cross in which just one trait is looked at. So on this slide, we are told that a heterozygous blue-eyed rabbit is crossed with a rabbit with pink eyes. What is the possibility of the babies being born with pink eyes? Let's isolate the first part, a heterozygous blue-eyed rabbit. We are looking at the trait of eye color, which can either be blue or pink. So let us choose the letter E to represent eye color in this instance. This rabbit is heterozygous, so it must be capital E and lowercase e. However, it is blue-eyed. So in a combination, or in a heterozygous combination, if it is blue-eyed, it means that the blue allele is being expressed, and so the pink allele is being masked in the heterozygous condition. So your capital E must be dominant, and it is thus blue in this instance, and by deduction your lowercase e must be pink. And when we look at our second rabbit, which is pink in terms of its eye color, there's only one genotype for that, small letter e and small letter e. If we were heterozygous, we would have blue eyes. And if we were homozygous dominant, for example, capital E, we would have blue eyes. So that's not true. And so the only possibility for pink eyes is small letter e and small letter e. In order to look at the second part of the question, what is the possibility of the babies being born with pink eyes, we have to complete a genetic cross. And a genetic cross involves a certain format which we have to follow. So we start off our genetic cross with our P1, or our parental one generation, which has the genotype uppercase E, lowercase e times smaller case e, which then undergo the process of meiosis to produce gametes. And if we think back to the concept of Mendels' law of segregation, our gametes from our first parent can be the dominant allele and the recessive allele, or it can be recessive allele and recessive allele from your second parent. Fertilization can then take place, and there is a possibility of the alleles meeting. And to look at that possibility, we draw a table of 3 rows by 3 columns, and we then cross the alleles in your gametes, as we can see. We then come to our F1 generation, which we have to express as a genotype and a phenotype. We can see from our table that we have 2 uppercase e's, lowercase e's, and that we have 2 lowercase e's. And in terms of our phenotype, a heterozygous condition will give us blue eyes, and a homozygous recessive condition will give us the recessive condition, which is pink eyes. So the possibility of a baby having pink eyes is 2 out of the total, which is 2 plus 2, which is 4, which simplifies to 1 over 2, which is then a 50% chance. So next, let us have a look at a dihybrid cross. A dihybrid cross looks at 2 traits at the same time. So let's read the situation on the slide. It says that tallness in snapdragons is dominant to dwarfness. So tallness is with a letter capital T, and dwarfness is a small letter t. While red flower color is dominant to white. So red is a capital R and white is a small letter r. Furthermore, and interestingly, the heterozygous condition of flower color results in a pink flower color. So normally, when we think about 2 alleles together, we assume that only the dominant will be expressed. But in this case, we have a condition in which neither allele is dominant, nor is neither allele recessive, and when they are together, or when they are heterozygous, they express themselves equally, and it is expressed as a mixture between the recessive and the dominant, and your recessive in this case is red, and your dominant in this case is red, and your recessive in this case is white, and the combination between red and white is pink. The question goes on further to say that a dwarf homozygous red snapdragon. So because it is a dwarf, it has to have the allele small t with regards to its tallness. And because it is homozygous red and red is dominant, we must have capital R and capital R. Is crossed with a plant homozygous for tallness. So if you are homozygous for tallness, you are homozygous for the dominant traits, so you are homozygous dominant, capital T, and white flowers. And if you have white flowers, white we know is your recessive condition, and in that instance you are homozygous recessive, which will be small letter r, small letter r. Okay, so I'm just going to erase these alleles that I've written on, just to make some space as we look into the genetic cross. So firstly, when we look at a genetic cross, we have to start off with our P1 generation, and our P1 generation is going to be our first snapdragon, which we saw was small t, large r, times by large T, small r. So T and R are the letters we use for two different traits. That makes sense because we are in a dihybrid idea. Meiosis takes place and then your gametes will be formed. In order to understand this concept of your gametes, you need to look at your first allele and then relate it to a possibility of the allele of your other genes. So in other words, you look at the first T, then at R, then at R, then you look at your second T, then at R, then at R, and you will find that your gametes will have the following allele. And the same applies to your second parent as well. We have to make the assumption though that both of these traits are controlled by a single homologous chromosome pair. Then with a dihybrid cross, we draw a table with 5 rows versus 5 columns, and the same thing applies. We then look at our F1 generation, represented as a genotype and then as a phenotype. And we find that in this instance, all our plants will be tall and they will have pink flowers. Pink means we are in the heterozygous condition, and that was told to us in our information, which we looked at the beginning. So next we come to the idea of blood groups as our third example of a genetic cross. And blood groups is an example of multiple alleles. And what this means is that we don't only have 2 alleles which can control blood groups in humans. We in fact have 3 alleles which can control it. There are 4 types of blood groups in humans: A type, B type, AB type, and then lastly we have blood type O. There are 2 possible genotypes under blood type A. We can have the following condition, or we can have this condition. So our first condition is a homozygous dominant condition, and our second condition is your recessive condition. And we use the letter I as it refers to the enzymes that are involved in the red blood cells of a blood group. The same can apply to blood type B. We can have it as a homozygous dominant condition, or we can have it as a heterozygous condition. Blood type AB has one possibility only, and this is an example of codominance, as we saw in our example with our snapdragons. We can have your two alleles interacting together, and this is an example of another type of this interaction in blood type AB, in which blood type A and B can be found together, and blood type O is simply your homozygous recessive condition. So keeping all of this in mind, we can have a look at a genetic cross related to blood types. So this question says, determine the possible blood genotypes of a child between a mother who is blood type A and heterozygous and a father who has blood type O. So I won't go through the entire genetic cross again, but we know that since the mother is heterozygous for blood type, this will be her genotype, and that will be crossed with the father who has blood type O, which has to be a homozygous recessive condition. We then undergo meiosis to produce our gametes. Fertilization takes place. We have a look at our table. We are looking at one trait, so it's a monohybrid cross, therefore it's 3 rows by 3 columns. And we then come to our F1 generation, and we can determine our genotypes and our phenotypes. Our last example of a genetic cross is what we call sex-linked recessive disorders. A sex-linked recessive disorder is an allele found on your X chromosome which does not have an allele on your Y chromosome. Examples of a sex-linked recessive disorder includes hemophilia, colorblindness, and muscular dystrophy. Because it is sex-linked, we have to look at your sex chromosomes. In our previous genetic crosses, we weren't focusing on X and Y because they weren't sex-linked recessive disorders. But because it is a sex-linked recessive disorder, the X and X or the X and Y combination become extremely important. So let's look at hemophilia to understand the ideas behind sex-linked recessive disorders. Hemophilia is a sex-linked recessive disorder, and it is caused by, let's say, the recessive allele small letter h. So there are 3 possibilities in a female. Firstly, a female can have X capital H, X capital H, which means that she is dominant for the idea of hemophilia, and hemophilia is represented as a recessive trait. So in this instance, there will be no hemophilia. Another possibility for a female is to have a capital on one X and a small on another X. However, the capital H will mask out the effect of the small h, and there will be no hemophilia. If a female has the 2 recessive alleles, she will then have hemophilia, because hemophilia is a recessive disorder. There's also 2 possibilities in males. A male can have either an X capital H, which will not bring hemophilia because XH is a dominant allele and hemophilia is a recessive disorder, or a male can have X small h, and that will definitely bring hemophilia because hemophilia is a recessive disorder. There is nothing on your Y chromosomes because, by definition, a sex-linked recessive disorder is a disorder found on your X chromosome which does not have a corresponding allele on your Y chromosome. So when we look at this concept on our slide, it makes sense to us why males often suffer greatly in terms of numbers compared to females when it comes to sex-linked recessive disorders. Males can either have or not have the condition, where females can not have the condition, mask the condition, or they may have the condition. So let us now look at an example of a sex-linked disorder. The question reads as follows. In humans, color blindness, and we know that color blindness is definitely a sex-linked disorder, so we will be dealing with XX and XY in this question, is due to the recessive allele small letter a, and normal vision is due to the dominant allele. What is the expected offspring between a normal man and a colorblind woman? Give both genotypic and phenotypic ratios. Show cross in detail. So let's start by looking at the man. We are told that the man is normal, and because the man is normal, he must have the dominant allele, because color blindness is caused by a recessive allele, and there is nothing on your Y chromosome. Your female is said to be colorblind. So when we look at the female, we can now say that since she is colorblind, she must have both recessive alleles. So this is the genotype of your female, and this is the genotype of your male. And so if we use our knowledge of monohybrid crosses, we can continue with this idea to answer the question. The key idea when working with sex-linked disorders is to always remember to add in your XX and XY, because they are disorders that are found on your sex chromosomes. So that brings our unit of genetics to an end. I hope it was worth watching and that you understand a little bit more about genetics. Thank you so much for watching, and I will catch you soon.